Welcome to this explainer on the microarchitecture of the human body. Today, we're unpacking a genuinely spectacular story. We're going to explore exactly how trillions of individual, totally isolated cells manage to construct the cohesive, incredibly resilient tissues of the human body and do it completely from the ground up.
Okay, let's dive into this because it's honestly an enormous biological challenge. Just think about the sheer mechanical tension required to hold a living organism together. You've got these individual microscopic entities that somehow have to withstand gravity, movement, and constant physical stress without, just tearing apart. So, how exactly does biology solve this massive structural problem?
To figure this out, we're going to treat tissue biology exactly like building a house. We'll look at four phases: anchoring to the matrix, linking cells to their neighbors, creating selective tissue barriers, and finally, direct cellular communication.
All right, phase one, anchoring to the matrix, laying the foundation. Before a cell can even think about forming a tissue, it absolutely has to establish a solid biological foundation. And the heavy lifters of this foundation, they're receptors called integrins. These are heterodimeric proteins, which just means they're made up of two different parts, and they physically span the cell's outer membrane. They reach right out into the extracellular matrix, that's the scaffold outside the cell, to bind directly to adhesion proteins like fibronectin, laminin, and collagen. This is literally the bridge between the inside of the cell and the outside world. Now, because integrins are heterodimeric, they're always composed of two different chains, an alpha subunit and a beta subunit. And, as we know, biology absolutely loves modularity. Out of 16 identified alpha subunits and 8 beta subunits, nature mixes and matches them to form about 22 distinct integrins. But what dictates what these integrins actually bind to? Well, the alpha chain features these specialized sequences with cation binding sites. Because the interaction with the ligand is cation dependent, these specific alpha subunit sites play a massive role in dictating the ligand specificity of the whole receptor. It's just a brilliant piece of biological engineering. And this brilliantly illustrates the concept. In a rat fibroblast on a culture dish, fluorescent staining lets us see the actin cytoskeleton glowing in green, and the focal adhesion kinase, which is where the cell anchors to the dish, glowing in bright red. You're literally watching abstract molecular concepts become real, glowing biological tethers right there at the border of the cell. But here is where we hit a really profound biological Goldilocks principle when it comes to how these cells stick. We actually don't want these connections to be too strong. If an individual receptor bound too tightly to its ligand, the cell would be irreversibly glued to the matrix, completely unable to move. Instead, its survival depends on multiple weak adhesions. By combining a bunch of weak affinities, a cell maintains enough sticking capacity to hold on, but it still retains the freedom to break those contacts and actively explore its environment.
Moving on to phase two, linking cells to neighbors, building the walls.
So epithelial cells are held together by these incredibly robust anchoring connections called adhering junctions, and there are two main types you need to know. First, the zonula adherents, which connect to the cell's internal actin filaments. And second, the macula adherents, more commonly known as desmosomes, which connect to intermediate filaments. If you want to visualize desmosomes on a macro scale, think of them like microscopic spot welts. They're exactly what allows tissues, like human skin, to withstand severe physical abrasion day in and day out without falling apart. Now, the primary proteins making these adhering connections happen between neighbors are caterins. Specifically, E-caterins act quite literally as molecular zippers. Extending across the gap between two cells, the caterins from one cell interact and tightly interlock with the caterins of the adjacent cell. They mechanically zipper the neighboring walls together, creating a unified, super resilient tissue layer out of individual units. Next up is phase 3, creating selective tissue barriers, weatherproofing the structure. This weatherproofing is achieved through tight junctions, or the zonula occludens. These junctions fuse the borders of two cells together, wrapping around the entire perimeter like a continuous waterproof belt. Transmembrane proteins from each cell interlock right across the space. To ground this in human anatomy, just think about your gut. Your gut heavily relies on these tight junctions to act as a selectively leaky barrier. It needs to absorb vital nutrients efficiently, while simultaneously preventing the uncontrolled diffusion of water-soluble molecules from leaking back out.
And finally, phase 4, direct cellular communication, wiring the house.
Now, what's really interesting about this is the sheer scale, or honestly, the lack thereof. Cells communicate directly across a gap that is unbelievably narrow, just 2 to 4 nanometers wide. Across this microscopic chasm, gap junctions form, coupling the adjoining cells both electrically and metabolically so they can function together as one highly coordinated system. The specific wiring allowing this communication is called a connexon. Formed by a group of proteins called connexins, a connexin from one cell aligns perfectly with a connexin from a neighbor, creating a continuous, open channel. But the cell is incredibly selective about what actually goes through these wires. They open and close in response to calcium and pH levels. And when they're open, they allow small molecules and inorganic ions to pass freely, essentially acting as a biological bouncer that strictly lets in anything smaller than 1,000 kilodaltons. The channel is rigorously sized to block larger, complex molecules like proteins, nucleic acids, and sugars from crossing over. So the crucial point here is this. Everything we've just covered isn't merely about holding things together mechanically. This microarchitecture is deeply fundamentally tied to the cell's very existence. Cell survival and cell proliferation absolutely require these dynamic interactions with the extracellular matrix. Without the matrix binding and that cellular crosstalk, the biological signaling pathways collapse, and the cell simply cannot survive.
Which leaves us with a truly fascinating and maybe somewhat terrifying question to wrap up this explainer. We've seen how meticulously nature engineers these foundations, zippers, belts, and wires to maintain total order. But if a cell's very survival is dictated by these strict architectural bonds, what happens when a cell chemically severs these connections, ignores the matrix entirely, and decides to just go rogue? The breakdown of this beautiful microarchitecture is exactly where some of the most dangerous human pathologies begin. Keep that in the back of your mind as you dive deeper into cellular biology. Thanks for exploring with me.